beginning to talk about the independent research projects. So first, so far we've talked a lot about population ecology, but obviously populations don't exist in a vacuum, even species don't exist in a vacuum, they interact with each other. And so these interactions are really important for shaping the community and also for shaping their population biology. So a few quick definitions. A community is populations of multiple species that occupy the same area and interact. And we often end up ignoring some species when we talk about communities. Communities of species that we normally don't always bother with when we're talking about community ecology. Predators have gotten quite a bit of attention because they're big and easy to see and they tend to have large effects on communities. Yeah, a lot of times we end up ignoring microorganisms like bacteria and parasites and all that stuff. Parasites are super important and they do have big effects on communities, but it's only been pretty recently that people started being concerned with those small things that are hard to find. We'll also sometimes narrow in our focus for somewhat practical reasons and just talk about assemblages. So this is a group of taxonomically related species that occupy the same area. So if we think about our community on a coral reef, we have fishes and we have corals in this picture. If we're talking about an assemblage, we might only focus on fishes in that community. So we're narrowing in our focus a little bit. Main community ecology is to understand how communities are assembled and how they function. And a lot of times community ecologists try to determine whether there's rules about how communities are assembled. So can we make predictions about community characteristics based just on the species that are there or the types of interactions that occur? So today we're going to focus on different types of interactions. Community structures influenced both by abiotic factors and biotic factors. We're really going to focus on biotic. And a lot of these we've mentioned before because they also influence population dynamics, they influence the evolution of different life history strategies, natural selection, things like that. So there's 5 main types of species interactions, and we classify them based on the 2 different species. So starting with competition, that's a negative interaction between 2 species. Both species are worse off because of this competition. An example of that are some surf perches. They've been shown to compete with each other and limit each other's distribution and sometimes also their growth. Predation and parasitism we group together.
Sorry, what's the name of those fishes?
They're surf perches.
Surf what?
Perches, surf. They're common in temperate ecosystems, including off of California, and we'll actually do an activity involving them later. Cool. So predation and parasitism we group together. This is when there's a positive effect on one species, a negative effect on the other. These are really easy to picture, so a fish getting eaten by another fish that's hiding in the sand, or these parasites that are attached on the side of this other fish. Mutualism is beneficial to both species. So one example that's been really popular since Finding Nemo is clownfishes and their anemones, where the clownfish provide nutrients for the anemone provides a shelter and a home for the clownfish. Commensalism is a +0 relationship, so only one species is getting a benefit, but the other isn't affected at all. So an example of that could be a predator that's hiding in a coral. The coral, it doesn't matter to it whether or not the predator is there, but the predator is getting a benefit because it's hiding and it's going to have an easier time catching its prey. So it's getting a benefit from hiding in the coral. Coral doesn't really make a difference whether the predator's there or not. And then finally, amensalism is when there's a negative effect on one species and no effect on the other. sometimes you could think of this as happening, damselfish protect their territories, and they'll sometimes chase away butterfly fish. So butterfly fish feed on corals. So the damselfish is chasing away something that actually wouldn't have eaten its algae, which is trying to protect anyway. So butterfly fish are getting a negative interaction. They can't get to their food, but the damselfish, not much is happening to it. So the cost of chasing it is very little, so it has a 0. And we'll go over some other examples of each of these interactions. So first, come into more detail. So this is organisms inhibit each other's access to mutual resource that is in limited supply. So the resource has to actually be limiting in order for there to be a negative effect on both species. So and the definition of a resource is any consumable biotic or abiotic factor required for survival, growth, or reproduction. So this can include things like food and shelter, space, and it can also include things like meats. So competition can occur both within and between species. Intra-specific is when there's competition within the same species. And this is a main source of density dependence, that direct density dependence that's regulating. And then interspecific occurs among individuals of 2 or more different species. So if we look at this picture down here, first, what kind of competition is occurring between the barnacle and the mussel? Yeah, interspecific, 2 species. And what about between these 2 mussels? Intra, same species. Alright, and then there's 2 different types of competition. First we have interference competition, which is direct competition. And this includes aggression and territoriality. So we have grunts that are actively being aggressive towards each other. They're comparing their mouth sizes, and that's how they fight. Or you can see hermit crabs fighting with each other for shells. So the way we draw this interference competition is like this. So the circles show the different species, species A and species B. The arrows that are solid means there's a direct interaction between those 2 species, and the minus signs means that it's a negative effect on that species. So these sorts of interaction webs are going to be pretty important as we go through this lecture. But then there's also another type of competition that can occur indirectly, and that's exploitation. And this is when species actually compete via another resource with each other. So you can think of barnacles competing for space. They're not actively fighting with each other. It's just whichever one gets to the resource first gets to have it, and anyone else doesn't really get to. So this interaction web is a little more complicated. First we have whatever the resource is. Both species A and species B have the same relationship with it, so the resource is positive for that. It has a positive effect on species A if it's space, for example. That species can have a negative effect on its resource, so if you think about eating prey, right, that's going to be a negative effect on the prey. And then because they're both having, they're both basically competing for the same resource, even if these 2 species aren't in direct interference competition with each other, they still have a negative effect. So that's the dotted arrows, means it's an indirect interaction. So it's a minus indirect competition because they're competing for the same resource.
When you say A and B, does it mean individual or species? It's
a species. Yeah.
So you can have but you can have 2 of the same species competing for the same resource, right?
Yeah. So individuals of the same species are pretty much always, most of the time, are going to be competing for the same resources. Occasionally they might not be if they're like different ages and they use different foods, for example, but most of the time individuals in the same species are competing. So these webs are really useful for looking at inter-specific competition and interactions between multiple species. Good question.
So if there was the same species, it would be like A and A.
Yeah, we usually don't draw it that way because you already know that A is encompassing the different individuals within that same species. So just by having the A, we know this is occurring. And we can pretty much assume intra-specific competition. So we mostly just use it for looking at interactions between or among multiple species. Cool. Anything else? Alright. And we're going to see a cool video real quick. So often we think of exploitation competition as more sessile organisms, but there's also examples like of corals that they engage in interference competition even though they're sedentary organisms. So, play that real quick. Greater the likelihood of competitive exclusion, which would lead to the extinction, or at least local extinction, of one of those species. And over evolutionary timescales, this ends up leading to resource partitioning. So species end up using different resources, which helps enable coexistence. Now this type of thing is more common to happen in really stable environments. So Gauss discovered this looking at different species of paramecium in the lab. So this is where it's just in a beaker. When you put the two species together, one went extinct. Basically, if he grew them separately, they always did okay. And what kind of growth curve is that one? Yeah, logistic growth. Cool. So in the lab, you can get these really clean logistic patterns too. So a little more about what this thing is. It's basically how a species fits into a community, and that involves its biotic and abiotic requirements and all of its tolerances for different environments. There's been a lot of definitions over the years, but the best known one is came up with it in 1957. It's basically the role the organism plays in the environment, and this can be determined by measuring all of that organism's activities and requirements. So, for example, some organism that lives in the intertidal might be able to exist in this zone, so in this range of zones in the height of intertidal, and it also may be able to exist in this range of wave exposures. So this particular one can live in basically mid-intertidal zones and mid-wave exposures. And it ends up getting complicated pretty quickly because you think of all the different factors that play into whether a species can exist there. So then you can also add in, for example, percent cover of macroalgae is another factor that might influence whether it can exist in that space, and it becomes a box. And pretty soon it comes into an n-dimensional hyperspace, so you can have axes going into any number of different dimensions. So there's a ton of different factors we can think of that fits into an organism's niche. And there's two main types of niches. So we have fundamental, realized. So the fundamental can basically be thought of as an organism's potential niche or its potential role, and the realized is its actual niche. So fundamental is determined by the physical factors and resource requirements, and this is what you would see if there were no other organisms around. Whereas the realized is determined by both the physical factors and resource requirements and the biological factors. So this is what is actually realized in the presence of other organisms. So that's how you can remember realized niche. So if you have a fundamental niche of this size, your realized niche is always going to be smaller or perhaps the same size as the fundamental. So usually things like predators and competitors end up limiting the realized niche to a smaller space than the fundamental niche. So a way that allows competitors to coexist is through resource partitioning. So if we have some resource gradient, this can be anything, it can be food size, it can be depth, height, and intertidal. And we see each letter in color represents a different species. We see right here there's a lot of resource overlap, so there's really strong competition among these different species. And what ends up happening over evolutionary time is a lot of times species adapt and end up slightly shifting their requirements. That way there's not as much overlap. So they basically partition the resources so that way every species ends up having access to something. And this is called species packing, when we have a ton of species all packed in close together with narrow resource gradients. Competition would cause a couple different types of spatial patterns. So again, if we think of space, a lot of times it'll end up being where competitors have non-overlapping distributions, so they'll be separated along that gradient. In the intertidal, we see this a lot with barnacles and mussels, where there's kind of a clear line where the mussel bed ends and then the barnacles begin. With rockfishes, we also have along reef depth. Some species exist deeper depths versus some species exist in shallower depths. And instead of being completely non-overlapping, competition is also manifested by a negative relationship in abundance between two species. So if we were to observationally go out and look for competition, sometimes we can see whether there's a negative relationship between two species. And this could be some evidence of competition. Some cases, this is a gradient. So you'll just have fewer of one species where there's a lot of the other, and then vice versa. And sometimes this ends up being more patchy or clumped distributions. Then if we were to get rid of so if we were to remove one of the competitors, we expect to see competitive release. So that's a change in distribution or something like growth in response to the removal of a competitor. So it's basically the difference when the two species are together versus when they're apart. So in this example, we have barnacles and mussels. When they're both there, there's this clear line about halfway through the tidal height. If we were to remove mussels, barnacles expand out into the rest of the tidal height. And in the absence of barnacles, the same thing happens for the mussels. So there's a competitive release when we remove the competitor, and that other remaining species is able to expand. So we could test for this either observationally by just going to areas that have barnacles but not mussels and mussels without barnacles, or we could test for it experimentally by actually removing each species. And which do you think would be better?
Experimental.
Yeah, experimental, and why is that?
Because they can move by itself, I guess.
Yeah, so we have more control, control of what in the experiment?
Of what is moving faster.
Distribution of it for where, which the
Okay, does anyone else help out with what we're controlling in an experiment?
Your environment, right? Well, you're controlling, like observationally, you don't know why there wouldn't be barnacles in a location, but in the lab, you know that they should be able to grow in your environment.
Yeah, exactly. So it doesn't necessarily have to be in the lab, it can still be in the field experiment, but we're controlling all those other variables in the environment that we don't care about. Because otherwise, if we just go to two sites and we see, oh, there's mussels here and barnacles over here, there could be some other factor that's influencing their distribution that's not competition. So the strongest test for competition and for competitive release is by looking at the organisms together and then doing a removal experiment and seeing what happens when they're apart. Alright, so I have a hint that will hopefully reinforce some of these concepts of competitive exclusion and niches regarding surf courses. So you can go ahead and work through this on your own or with your partner, and then we'll go there. So we don't have data for these points, so it's a little bit sketchy to interpolate between where you have the data, because we don't actually know. It might have just, these lines were just drawn straight in between the points, but it might be that really there's none starting at 4, or there's none starting at 5.9. If we wanted to be more thorough, we would sample all of these different depths and fill in the gaps. Yeah, so I'd be wary of concluding things where there's not data.
So would you assume for the, when you remove the block search, that they're only at 3 to 6 and nowhere in between 3 and 6?
It seems most reasonable that they are also in between those two depths, but I guess we can't know for sure. But usually species will be in a depth range rather than being really shallow and then nothing, then there again,
So why not use a bar graph rather than this?
Yeah,
Because wouldn't it make more sense if you're not, if you can't conclude that then?
a bar graph for these data would be better. I would agree with that.
Be a
So
lot easier.
you can write to the authors of this original paper that I stole the figures from.
Yeah.
So if you had more than just 3 depths and you're able to fill in some more, then this type of graph would be great because you can really clearly see the trends. But because it seems like they probably treated depth as a categorical variable, because that's all they could really do with 3 points, you're right, I think a bar graph would be better. So, take it up with them.
After you get the copyright issue taken care of.
Yeah. I think this paper is from the 80s, so. Yeah. Alright, so what about for black perch? What depths are they found in the control with the competitor present?
Across all depths.
Yeah, all the depths, 3, 6, and 9. And then what about when the competitor is removed, where are they found?
They found across all depths, but with increase in depth 3 and 6, more increase in 3 and 6.
Yep, so they're still found across all the depth range, but now they've had an increase in density at least 2 of the depths within 6 meters. Cool. So, going back to the striped sea perch, is the realized niche and the fundamental niche the same?
No.
No. Cool. Which one is smaller? The
The
realized.
realized.
And why do we think the realized niche is smaller than the fundamental niche?
Because in the presence of a competitor, they are limited in numbers.
Yeah, so it's competition, interspecific competition that seems to be limiting them. And it's, but in the absence of it, they're able to actually expand their depth range into new areas. So their fundamental niche is bigger than their realized niche.
Would you explain one more time the fundamental niche and the realized niche?
Yep, so the fundamental niche is basically where or under what resources or what circumstance a species is able to exist without any other biotic interaction, so without predation or competition. Whereas the realized niche is where that species is actually able to exist once you include in those other interactions. So, the realized niche encompasses everything that affects the fundamental niche, but now we're also looking at their interactions with other organisms, so biological factors.
So in that case, the realized would be the open cycle?
So, let's see, black perch. So in this case, the realized niche out in the normal environment is gonna be the control, because that's what's realized in the presence of the competitor. But the fundamental niche, if we were to remove competitors, and theoretically we would also have to remove predators and other things, but that would be more like the open circles. Yep. Cool. Any other questions about that one? So then what about for the black perch? Is the realized niche and the fundamental niche the same? heard some good
It-
debates
it's-
over this.
Is the same if you're like looking at the depths only, but if you're looking at the density, it's not the same.
Right, so the niche is really concerned with just the depths, like presence versus absence. So in that regard then, is it the same?
Yes.
Yes, cool. But then is there evidence for competitive release? We'll start with one we're talking about, in black perch.
Yeah, because the density increased.
Yeah, so we still have some evidence for competitive release because there's an increase in density, but the niche, it's still occupying that same space on the resource gradient. So it's still there in the same depths that it was before.
So you couldn't use like the difference in niche as competitive release, like as the reason for competitive release. Like you could have the same niche and still have competitive release.
Yeah, because competitive release can be manifested in a lot of different variables. So it could also be growth rate, for example. It doesn't just have to be. It's not just presence absence. And then is there evidence of competitive release for the striped sea perch? Yeah, so that we see competitive release both in terms of its niche expansion, so in this case it's depth expansion, and also in its increase in abundance at least 6 meters. Cool. Yeah.
I think I saw that, like, on the one on the north, like, if you take out the other guy, then the density is increased, so their fundamental niche drops.
So they're yeah.
So this, okay, so something, niche is just depth, is area.
Yeah, it's just where they're found. Let's go back to this. So if we think about the niche, if we just think about it on this axis, so wave exposure, think of this as depth. We're just looking at where it's found, but this box says nothing about density, right? It's just the areas where it's present. Yeah. So for niche, you can think of presence absence. For competitive release, you could look for evidence in terms of presence absence, but also in terms of density, growth rates, any other like measure of fitness or performance. Any other questions?
Could the competitive release though be something other than its competitor? In terms of like, if we were to make the assumption that the population or the density of the black surf perch went down, or vice versa, what happens if that's not due to them being together or something?
So that's why.
That's the competitive release.
So that's why we do an experiment. So in this case, we actually, I didn't do it, but someone else. It wasn't just looking at areas or times when they were present versus absent. They actually were like paired sites and had them both present. And then in an experiment where you actually manipulate a factor, then you can say it's caused by that. So that's the benefit of the experiment, right? Is the causation. You're right. If we were to do an observational study, we'd have maybe some suggestive evidence for it, but we wouldn't be able to say it was definitely caused by competition or lack of competition. Cool. Anything else? These are all really good questions, so awesome. All right. One last thing to say about competition, and then we'll move on to the other interactions, which there's not as much to say about. So another way we categorize competition is based on symmetry or asymmetry. So we're looking at the relative competitive strength of the two different species we're competing. And when there's asymmetries, you have a superior competitor and an inferior competitor, or the same way to say that is a dominant competitor and a subordinate competitor. So in symmetrical competition, each species is having the same negative effect on the other. So the effect of species A on species B is the same as the effect of species B on species A. In asymmetrical competition, you have one species having a greater effect on the other. So in this case, the effect of species A on species B is greater than the effect of species B on species A. And we designate these relative magnitude of effects by the thickness of the arrow. So when it's a really thick arrow, that means that it's a stronger interaction than a thinner arrow. So in this first case, A is the dominant or superior competitor. Its effects on B are greater, so A is greater than B. Or in the reverse case, we have the effect of species B on species A is greater, so B, it would be the dominant competitor in that case. So how would you assess whether competition is symmetrical or asymmetrical, and which species is dominant?
Experiments.
Yep, so we'll do experiments again, because that's the best way to assess competition. Why don't we look at an example, and then we can decide. So in this experiment, we have two different fish. We have Degassi partitus, the bicolor damselfish, on the right, the one that's two colors. And we have Coco damselfish, Degassi varibalis, on the left. So again, they did a removal experiment. So the graph over there showing bicolors, this is the net change in density on removal reefs. So this is comparing the density of bicolors in the absence of their competitor, and it's relative to their densities on control reefs where they were both present. So the axes are different than we looked at here, but basically 0, if the points lie along 0, that means that there's no difference between the reefs with the competitor present versus the competitor removal. The points that are above 0 show there's an increase in density on the removal reefs compared to the control reefs with both species present. And then negative would mean that they did worse in the absence of the competitor, which doesn't really make sense. So if we compare these two, what do you see for the bicolor? What happens to them when their competitor, the Coco damselfish, is removed? Yeah, their density increases, right? And what about for the Coco damselfish? What happens to them in the absence of their competitor? Yeah, there's not really much of a change, so it mostly overlaps 0. So would you call this competition symmetrical or asymmetrical? Asymmetrical, cool. And which one is the dominant competitor?
So that's
This one is the dominant, cool, because this one is having a bigger effect on the bicolor than the bicolor is having on this one. Alright, and so finally, responses to competition. For all of these interactions, we can have individual responses and also population level responses. So individual responses, competition can change their behavior in terms of feeding rates or foraging distribution, can change physiological parameters. So we talked about growth rate. It can also affect reproductive rate. And it can affect morphology, so body size or biomass. And competition is a negative-negative interaction, so we'd expect all of these things to decrease in the presence of competitors. In population level responses, we can look at abundance or density. We can look at distribution or zonation. And we can look at overall demographic rates of the population, so population growth as opposed to individual growth rates. All right, any questions on competition? Cool, let's talk about predation. So the consumption of all or part of one organism, which is the prey, by another organism, which is the predator. So if you think of a diagram, here's a food chain. Again, this is all similar. So we have the predator, species A, has a negative effect on its prey, species B, because it's consuming it. Species B has a positive effect on species A, so the prey has a positive effect on the predator, right? The predator gets benefits from eating food. So you can have predators eating herbivores, you can also have herbivores eating primary producers, so plants or algae, and again, it's the same thing, where the herbivore has a negative effect on the plants have a positive effect on the herbivore. You can also make it more complicated and make it into a food web. So here we just have one predator, we have 2 different herbivores, and we have 3 different plants or algae. So in this diagram, the key points to remember are that predators and prey are on different trophic levels, right? So they're above and below each other on this. Within a trophic level, that often ends up being competition. So if we think of these 2 herbivores, they're eating the same food resources, so there's probably some sort of exploitation competition going in here. So predation is between different trophic levels, and competition is usually within the same level. And usually when we think of predation, we think of the crazy, scary things like that, but there's a lot of different kinds of predation, right? So it can be where you're eating different fish, or also like zooplankton, like whale sharks and humpback whales eat. They're filter feeders. They're still eating another animal, so it's still carnivory. Herbivory is another form of predation. This usually, often they don't eat the whole plant or the whole organism, so that's where that consumption of all or part of an organism comes into play. And then parasitism is a predation. So in this case, the consumer usually doesn't kill its food, and it also usually only feeds from one or a few different hosts in its lifetime, but it's still having a negative effect on its host. And we started to classify different types of effects on predators in terms of consumptive versus non-consumptive effects. So we used to only think about the times when a predator actually eats its prey, which is the consumptive effect. That's also called density-mediated interactions, if you want to be really jargony, but we don't have to do that. And this is direct losses or removals of individuals from a population. And this results in death of individuals, so it's directly affecting the mortality rate of the population. Non-consumptive effects, which are sometimes called trait-mediated interactions or risk effects, that's when there's an influence of the predator on a variable other than death or mortality. And this can be in terms of behavior, physiology, or morphology. And so recently in ecology, we've realized that non-consumptive effects can be as important, if not more important, than consumptive effects. So sometimes just the predator being there can change, for example, the growth rates of prey, and that still will in turn lead to population level changes. So an example of non-consumptive effects, here we have tiger shark abundance on the x-axis, so there's either low abundance or high abundance. This was an observational study, and they took advantage of the fact that there's seasonal differences in tiger shark abundances. So in this area of Australia, tiger sharks come into the season and they migrate out of the bay in the other. So that's how they got this gradient of abundance. And then they looked at the time spent feeding and the relative interior use. Interior areas of seagrass beds tend to be safer than exterior areas. And they're looking at these for a prey of tiger sharks, which are dugongs. They're basically like manatees. And you can see that just having the tiger sharks around in the bay, the dugongs greatly reduce their amount of time spent feeding and their amount of time using the interior areas of the seagrass beds. And another example where there's actually an experiment, in this case, they put out a model of a predator, so this isn't even a real predator, it's a fake predator. And they put that near some seagrass, and they wanted to measure the foraging rates of herbivores on the seagrass. And again, here we have a decoy control, so this was a decoy that wasn't shaped like a predator. We have control, so without any model or decoy present, and then we have with the predator decoy present. And you can see that there is a huge reduction in the consumption of seagrass in the presence of this model predator compared to the two controls. So sometimes animals just, being afraid basically is causing them to decrease their consumption rate. So again, we have our individual and population level responses of predators on prey, and they're pretty much the same as for competition. So you can have changes in behavior, physiology, or morphology, and then you can also get eaten, either completely or partially. And population responses, again, it can change the abundance or density, their distribution, and it can also change the structure of the population. So a lot of times it's certain age classes, for example, like in fish, small fish are more likely to get consumed by predators. So if there's a lot of predation, you might have fewer small fish in the population. Same with age, sex ratio, genetics, spatial, all of those things can change based on which specific individuals and predators are eating. And as we saw before, predation can influence population regulation. So it's one of the ways that can impart direct density dependence, which can lead to population regulation of the prey. Okay, so we can also have complex interactions between predation and the other processes. So the most typical example is predation and competition often interact with each other. So sometimes in this example, if there's a predator that specializes on barnacles but only lives in the mid and lower intertidal, with predators, barnacles are restricted to the higher intertidal heights. If the predator is removed, then sometimes this can enable barnacles to extend further down, and they might actually be able to outcompete mussels. So another type of competition that's not true competition, but it manifests itself similarly and is mediated through predation is apparent competition. So in this case, we have species A and species B are both prey of species C, and whenever there's an increase of either prey, it leads to an increase in the predator, right? So if there's more prey around, the predator, its population can usually expand or increase in density because it's able to eat more prey. So this can increase the density of the predator, and that in turn can increase its predation on the other prey species. So this leads to a negative indirect effect of these two prey species on each other. And then we can also look at trophic cascades. So this is across multiple levels. In this case, we have a predator consuming its prey. So we have predator eating an herbivore eating its resource, so usually plants. In this case, this top predator has an indirect positive interaction on the plant, right? So because the predator reduces the plant's predator, and so that would enable the plant to increase. This interaction is indirectly positive. So whenever there's an indirect interaction, we designate it by the dashed lines as opposed to the solid lines. So can anyone think of an example that we've already talked about that's a trophic cascade? The first class. Yeah, the sea otters. Cool. So in that case, we had the killer whales eating the sea otters eat the urchins eat the kelp. Once the killer whales started eating more sea otters, they were reduced in abundance. That led to an increase in the urchins and a decrease in the kelp. Alright, any questions on predation? Cool. Commensalism, that's a one-way interaction where one organism has a negative effect on the other. So that looks something like this, where there's really actually no interaction there, negative interaction the other way. Another example of this is a stingray unburying non-prey invertebrates while digging for food. So they'll go along, they're digging up their own food. It doesn't affect the stingray at all to dig up these other species, but once it digs up these other species, they're more likely to get eaten. So the stingray is having a negative effect on those other invertebrates. Those other invertebrates are having no effect on the stingray. And then mutualism or commensalism. This can occur within or between trophic levels, but usually it's between them. Mutualisms, there's two main types: obligate, where the two species are actually required for each other's coexistence, so they can't exist without them. Facultative, they're not required, but it's still positive interaction. And in terms of spatial distributions, we'll often see the opposite as what we saw for competition. So as the abundance of one species increases, the abundance of its mutualistic partner will also increase. Commensalism is also called facilitation, but in this case, only one species is having a positive effect on the other, and the other has no effect on the first species. So can anyone think of any tests you could do to distinguish mutualism from commensalism?
Separate them, right?
Yeah.
See which one survives better.
Yeah, you can separate them and then measure some sort of response, whether it's survival or other response like growth rate, and see whether there's this two-way interaction or it's just one way. We're running out of time, so I'm not going to show this video, but if you want to look at it later, it's pretty cool. One example of a mutualism on coral reefs are shrimps and gobies, where shrimps are completely blind, but they'll dig out these burrows, and there's species of goby that will sit at the burrow, and they gain from the shrimp protection from predators. They can dive into the burrow, and in return, they actually warn the shrimp when a predator is coming. So the shrimp can't see. The goby will actually wag its tail, and that alerts the shrimp, okay, don't come out of your burrow right now. So that's an example of a mutualistic relationship. They're both beneficial. Some other examples of mutualism and commensalism. We already talked about anemone fish. Interactions is one as well, and this one is an indirect mutualism. So instead of directly benefiting each other, in this case, the interaction is operating through a third party. So kind of like that exploitative competition where it's operating for a resource. In cleaning mutualisms, we have cleaner fish. That are consuming parasites that live on the outside of other reef fishes, their hosts. Right, the parasites have a negative effect on their host. Host has a positive effect on the parasite. And because of this interaction, where we have cleaner fish are negatively affecting parasites, parasites are negatively affecting the host fish, leads to an indirect positive interaction between cleaner fish and host fish. And it works both ways, because the cleaner fish are getting food from the parasites via the host fish. So it's the idea that basically the enemy of my enemy is my friend. I'll leave that up for a while too. And then the last example is commensalism. So there's algae that grow on turtles. They basically get a free ride, then access to space and light and nutrients on the turtle shell, but they don't really affect the turtle very much at all. So turtle isn't getting any benefit from the algae, but the algae isn't harming it. All right, any questions? So So why don't we take a 10-minute break, you can go relax, whatever, and then come back to the same room and we'll start going over science.